Time to start. We have with us today David Buchwalter. He's going to give us a seminar in association with his being considered for promotion to full professor. David received his PhD from Oregon State. It's here that he started working with aquatic insects, which really has been his model of choice throughout his career. And I'm sure we're going to hear more about that. Upon gaining his PhD, he went on postdoctoral studies at the U.S. Geological Survey in Menlo Park, California. Here he was working with Sam Luma, who is a prominent ecotoxicologist, and David continued his work with insects studying the bioaccumulation of metals. He came here at State in 2005. He was hired as an assistant professor in the toxicology department. In 2008, he took on responsibilities for coordinating the environmental toxicology concentration within the department. David's interest from a translational standpoint really involves the bioaccumulation of trace elements, particularly as related to fossil fuel extraction, so mountaintop removal, fracking, things like that, resulting in the contamination of streams and exposure of organisms, and with his interest, aquatic insects, to contaminants from these sources. From a basic research standpoint, his interests lie really in the physiology of the uptake, the accumulation, the elimination of trace elements. He's interested in species differences as taken from an evolutionary perspective. And he's also interested in environmental factors such as climate change, what these changing environmental conditions can have on the dynamics of species interactions with trace elements in their environment. Students enjoy working in David's lab because they get to take trips to the mountains to collect their species, but in recent years, David has, for the most part, domesticated a species. He has a species of mayfly that he can work with in the lab throughout its entire life cycle, and it certainly has significantly advanced not only his work, but really the science in general with respect to studying aquatic insects in controlled laboratory conditions. And I expect we'll hear more about that from David. The title of his talk is Linking Physiology and Ecology in the Changing World.
Thank you, Jerry. Can everyone hear me okay in the back? I'm going to sort of do this in a kind of a biographical way, just to get a sense of how my thinking and sort of research has, my way of viewing it has changed over time. So I'm a freshwater biologist, and since the other two speakers started with the big picture of the planet Earth, I decided I shouldn't. But basically, if we look at the planet Earth, it's, most of the water that we have here is salt. We only have a little bit of fresh water. Most of that is sort of unattainable. It's either locked up in the poles or is deep underground, and so this is like this limiting resource in a lot of ways. And so we obviously, as people, have these huge demands for fresh water, whether it's drinking water, food production, energy production, or other industrial processes. We just use water massively, and we change not only the quantity but the quality of water in a big way. And you could argue that our demands for fresh water, or our species, are unmet. We have lots of people dying every year from water-related diseases, and we also have huge areas in the world where there's not enough water to grow food, and people are living in famine conditions. Even here in the United States, I did my postdoc work in California, there's not enough water in California for all of the needs and demands there. There are streams that are purely wastewater. There are massive relocations of water. Rivers don't flow into the estuary anymore. They get pumped back up into the agricultural valleys. So we are sort of struggling with how we're managing our water. And despite the fact that we have this limited resource that's not really meeting our needs, we actually do really bad things to it. So not only are we using it, but when it returns to the environment, it's often in a way worse condition than when it started. And so we're sort of failing in a big way on how we deal with water resources. The other side of the freshwater, sort of going not from a human-centric way of looking at things, is that these ecosystems are disproportionately important in terms of maintaining biodiversity. As a footprint, freshwaters make up about 1% or less than 1% of the Earth's surface. They support about 10% of the Earth's biodiversity. And all accounts are that in this modern era where the world is changing very quickly, freshwater organisms are hurting more so than terrestrial and marine organisms. These are the systems that are the most changing the fastest. Now, this biodiversity in freshwater is really driven by primarily one faunal group, which are the aquatic insects. And so these things, if you go and sample a system like this, especially flowing water, streams, and if you go ahead and sample what's on the bottom of these streams, typically like 95 to 98% of the specimens of things that you find there are going to be insects. In terms of like the overall biodiversity, these are different classes of aquatic just invertebrates, but you know, the insects dominate over all of them. So I was trying to sort of come up with like a very simple schematic that sort of describes my research overview, and it's something like this. So basically, each of these little jelly bean-looking things would be a population of organisms in a place, and that there are some agents of change that occur, and then what you get left with is this highly simplified, you've lost some biodiversity. So you've lost some species, but you've retained some. And really, from day one, I've been interested in this question of like, how does the physiology of these organisms, or the differences in physiology of these organisms, affect or determine this differential susceptibility? So I've always been interested in this concept of why does this one persist and this one goes away? Like, what drives that? And so it's really this marriage of sort of ecology and physiology. The patterns out here this is largely an ecological question. Understanding what drives those patterns is largely a physiological question. I'm trying to sort of engulf both of these things. So in my early days in Oregon, I witnessed the rapid evolution. This is a toss out to Brian of my school's logo. It went from a cheerful, happy beaver to a more warlike, angry beaver. But I was also spending a lot of time on streams and rivers as a fly fisherman, and I don't really consider this wasted time because I wouldn't trade it for anything. But I developed something of a natural historian sort of perspective on these organisms and the environments that they come from, which I think is really important to have some understanding of the biology of your organisms, the timing of when they're around and when they're not around, the things that happen and make some of them go away. And at this time, there was no labs in the United States doing rigorous lab-based science using stream insects. Occasionally, an ecologist would get an idea to do an experiment in the lab. Yeah, not great results in terms of good physiological rigor. And so there was really nothing happening here. There were no labs to go to if I wanted to do physiology. But at the same time, these ecological monitoring programs were increasing nationally. States, all under the Clean Water Act, have the obligation to monitor streams. And there are federal programs and state programs. And so all of this is driven by the Clean Water Act, which sort of historians tell us was largely driven by people just being fed up with our waters being treated in a really bad way. And this event, the kind of the river catching on fire, is sort of, when you read the history of the Clean Water Act, they always talk about this event. This river caught on fire lots of times, and the last time it did, people were like, okay, we need to do something better about how we're managing our waters. And so this law created this ecological monitoring infrastructure. We had many ecologists out there describing patterns in nature with really little to no support from lab scientists to help elucidate mechanisms of why certain species are going away, or even to diagnose the causes of ecological impairment. So these biologists could go into a river system, take samples, hey, the biology is pretty bad here. The next thing you want to know is, okay, well, what's causing that? If you look up in the watershed, you don't know if it's that power plant, that agricultural system, whatever, right? So there's no diagnostic capabilities because we don't know enough about the way the organisms work. And so that's kind of how I visualize what I should be doing, is like trying to learn about how these animals work from a physiological perspective. And so, again, this part of it, the challenge of this part of it is really done by other people. This is the these are the monitoring programs. That's not my problem. My problem is that there's like 8,500 species described in North America. So I'm going to start this research program from scratch, doing comparative physiology where there's like way too many of them, right? So a bit of a fool's errand. Maybe it still is a fool's errand. And so it's useful in thinking about these organisms to have something of a historical perspective about them. And so basically we think life on Earth, plants, 465 million years ago, first fossil evidence about 450 million years ago, first insects. Then we have a bunch of different extinctions, and then basically right up here is where we have us kind of separated from our earliest common ancestor. So where do the aquatic insects fit into this? Basically numerous invasions of freshwater habitats by terrestrial ancestors diving into exploiting freshwater habitats over evolutionary time. So there wasn't one invasion and then a bunch of speciation. These are not a monophyletic group. These are completely separate lineages, right? And so, like marine mammals, these terrestrial animals dive into water, make all these changes to their morphology and the way they live. The insects kind of do the same thing. And two of the systems that I'm most interested in are how they breathe and how they osmoregulate. For example, for breathing, there's a whole lot more oxygen in air than there is in water, right? And if we look at what terrestrial insects do, and presumably what the terrestrial ancestors of our aquatic insects do, they basically have this tracheolar system. This would be a spherical, this is like facing the outside world, this little muscle that controls the opening and closing of these basically pores, which feed these trachea to tracheoles, and these things end blindly on muscle fibers and nerve cells. They don't use blood to transport oxygen. Oxygen is transported as a gas through these tubes, right? So how do aquatic insects breathe? Lots of different ways. Some of them maintain that same thing. They maintain, like this one has one functional spherical way up here. It basically has a snorkel, right? And it has to maintain some contact with water on a periodic basis. We've all seen mosquito larvae wiggle up to the surface, grab a gulp of air, right? But many of the aquatic insects basically have completely closed themselves off. They're no longer having any functional spiracles. They've popped out where these tracheoles would be, where the spiracles would be. Now there's gills popping out of these guys, right? And so you have these animals that are completely dependent on dissolved oxygen. These are highly permeable surfaces, right? And there's a huge variety across the insect world of what they look like. Here's a gill of a mayfly, and these are all gills, but plates with fiber, these more sort of fibrous things on top of them. These structures are so conspicuous that taxonomists often use them to determine who's who. These are like big, important morphological characteristics on them. And so two good questions that I had as a graduate student were really to the historical or evolutionary solutions of facing that challenge. Okay, you're going to go from this from this terrestrial to this aquatic way of living. Does the way that you solve that problem do anything to sort of your susceptibility to new and novel stressors, like things like modern chemistry, like pesticides, for example? Or asked a different way, can susceptibility be linked to traits, physiological or morphological characteristics that would help me take this crazy, daunting amount of biodiversity, 8,500 species, and make it a little bit more tractable, like maybe big gills or no gills, or can we start lumping animals together based on some other things? And so, as a student, I was really interested in sort of identifying where on the body surface is there exchange epithelia. So here, example is the underside of a stonefly, and these are gills. These are sensory cells on the antenna of that same animal. And here's this another example of just lots of gill surface area. And my idea was that there was going to be potential for widely different pollution exposure among co-occurring species. You got a bunch of species in the same place. Some of them have these big available epithelial surfaces. Some of them have none of that. right? So if the chemical washes through, do some animals have more of an exposure to contaminants than others, essentially? And so we did this work using radio tracers. I worked with a labeled organophosphate insecticide, and I used tritiated water as a measure of how quickly they turn into body water. And we showed that species with different respiratory traits had very different exposures. So even though they're in the same water, internal concentrations of pesticides were much greater in animals that had gills and didn't have gills. So that was the sort of start, right? So did enough there to turn it into a postdoc in San Francisco Bay Area. I actually lived on this park, on Westwood Park. But this was really a great opportunity for me because I worked with a gentleman named Sam Luoma, who championed this idea of biodynamics. And this paper was published in 2005, and its last time I looked, it had 585 citations. Pretty important paper in our literature. But the idea here is that if we want to understand differential sensitivity among species, it's based on knowing something about the functional ecology of these organisms, something about their physiology, and something about the environmental chemistry that they're living in. And so these things like uptake from food, uptake from water, loss rate constants, these are things that I've done before using radio tracers, right? I had some experience as a master's student thinking about environmental chemistry and metals. I had a little bit of this background of just knowing the natural history of my animals a bit. And so basically was able to take this and put it together in my research program as a postdoc. And I was able to take advantage of the fact that the U.S. Geological Survey had this project up here in Montana where here the Anaconda Mine was the biggest copper mine in the world, operating from like early 1800s all the way into mid-1900s. They also had a smelter, so not only would they have issues associated with mining, but they had issues associated with smelting. The stack is a really prominent sort of aspect with the landscape, and people downwind of the smelter complained about with health problems. They were called downwinders. But this also, these plumes of metal-rich pollution basically settled down and contaminated this floodplain for 100 and something miles. So it's like the biggest Superfund site in the country. And so what we have is up here, nasty polluted, and then as you go downstream, it gets better and better and better. People actually catch trout down here. It's not completely hammered down there. But there's a few different things that are kind of key here. One is that you have this gradient of metal pollution and you have accompanying species loss. Up here you can only find 1 or 2 insect species. Down here you can find about 60 or 70, right? And as you go along the way, you lose more and more species as you go, gain more as you go depending on treatment. And then if you take, okay, let's just look at one site. It doesn't really matter where you are. If you just pick one site along the river, sample the bugs that were there, separate them into species, and then measure their contamination loads, wildly different concentrations in tissues. So even though they're in the same water, living in the same location, these organisms are massively different in what they're acquiring. And so finally, one last bit is that there was also some dissonance here because while metal effects in the field were obvious, traditional toxicity tests were not predicted. The normal way of doing tests, bring these animals in the lab, give them short-term exposures, were resulting in toxic values that were basically would say that you need more metal than that to make these organisms sick. But in nature, you can see clear effects. So there was this, and I'm going to circle back to this idea later in the talk here. And so now, rather than thinking about gills as the traits of interest, now we're thinking about differences in the way that they manage their salts and how they osmoregulate. And so basically here, the idea is that the ancestral state of these guys is doing everything in their GI tract. All water-salt balances in the GI tract. There's no structures on the body's surface that do any of that job. The aquatic solutions vary a lot, and they include the expressions of ionocytes, or what are called chloride cells, or chloride epithelia on the body surface, and these things are very busily taking up metals from the surrounding environment. They're basically taking up salts, but metals are utilizing those transport processes. And so these structures vary. Here are ionocytes in the base of a gill in a stonefly, for example. Here's one up close. These are mitochondrial-rich cells. They do there's a lot of pumping going on here. In more derived forms, you have these patches of epithelia on the abdomen here, for example, in this caddisfly. In mosquitoes, you have what's called an anal papilla, basically a dedicated organ that's involved in salt exchange. And so the structures are different, the relative amounts of it, the relative rates at which they're moving ions are radically different, and we wanted to explore this in the context of differential metal exposure. And so again, relying on radio tracers, using isotopes of cadmium and zinc this time. But these isotopes are different from the previous ones. These are things like gamma emitters. They're better because they allow us to non-destructively assay the animals and then return them to the exposure again. And so what we can do is we can do a lot of more refined work with rates and kinetics. And so, for example, we take these animals out of a clean environment, all exposed to the same water. Panel A, we're looking at the accumulation of zinc over time in different species. Here, we're looking at the accumulation of cadmium over time in a bunch of species. And basically, they're radically different. Even though they're in the same exact water, their rates of acquisition are very much different from each other. And so, after working on this sort of question here in 2005, life gets pretty real. I get this job in July, moved here with a very, very pregnant wife. Began working here in August, had a baby in September, and then onward, from October onward, begin to realize that there pretty much is no EPA money to do any of this, and that NSF biology is really not interested in toxicology. And so in terms of like, how am I going to keep my program floating, or even just establish a program, but was sort of like, yikes. But I also read a paper that really heavily influenced the way that I was thinking about my work and the way that I could package my work. And that paper was this commentary by Ted Garland Jr. and others, basically championing the idea of using phylogenetic approaches in comparative physiology. And so I was doing comparative, I was comparing across species, but I wasn't applying the appropriate framework in my mind to do comparative work the right way, essentially. And so one idea basically is that we're all biologists studying species here and now for the most part. But what they are is this function of history, right? And as we look for patterns, like sometimes those patterns are determined a little bit by this history. And one thing that tends to happen is that closer relatives tend to look alike, right? It's sort of like this ubiquitous thing. And so, hummingbirds look like hummingbirds, and they don't necessarily represent independent pieces of information, right? African elephants, Indian elephants, pretty identifiable. But then you've got these relatives, the hyraxes, which is like, okay, they're in the same clade, but they're very different, right? And you got problems of sometimes, even in the same species, traits can vary widely, right? And so the question is, okay, we're working across these, working across species, we want to know something about the phylogenetic patterns associated therein. And so if you've got data where you've got, species in columns and some observations in columns, if you do any normal statistical thing where there's a correlation or regression, you're making the assumption that all these species are independent. You're basically assuming the special case phylogeny. You're assuming the star phylogeny, where the reality is that your organisms are related to each other in some crazy hierarchical fashion, and using phylogenetic and appropriate statistics takes this into account, and actually allows you to even ask different questions than you could using conventional methods. So one of these statistics is basically quantitating the strength of what you're measuring, how tightly aligned are your measurements to phylogeny. Right? So the idea here is that while the tendency for related insects to resemble one another is ubiquitous, it's not always true, right? So we want to know, like for the traits that we're interested in, are they tied to phylogeny or not? So in this case, we've got the same organisms, two different traits. This is lizards or something, skinks, optimal body temperatures for sprinting, There's really no apparent trend with phylogeny at all. But if you look at over here, the preferred body temperatures, then you see this real partitioning that like preferred body temperatures are very much a phylogenetic thing, right? And so the statistic K describes the strength of this phylogenetic signal, essentially. And so here we have a low K value, insignificant P, and you do this with this randomization test. You basically take your data, you plot it onto the trees, you calculate the variance of the data in your trees, and then you scramble your tip data, the data, your species data, keep the tree the same, you scramble your data. You do that like 1,000 times. You get a world of possible fits of variances of your tip data to your tree, and you can basically put a p-value on it. And so basically now we're going back to this like Llewellyn rainbow concept of differential sort of susceptibility. These things related to bioaccumulation, these things related to the way the animal deals with that bioaccumulation that ultimately blend together to come up with differential susceptibility. My question was, to what degree do these individual traits follow phylogeny? So I'm basically taking the Llewellyn rainbow idea, putting it under this Garland et al. Idea, and seeing how well they map. And so I contacted Ted Garland. I had the beginnings of this data set, and then I think I had 11 species or 12 species. It's like, go away, come back when you get 20. And so we spent the next couple of years measuring with isotopes uptake rates from solution, elimination rates, and aspects of detoxification in all of the animals that we work with. We put it all together, and so basically here's our data set, and this is the raw data. But here we have an uptake rate constant. Basically, over here you're fast at picking something up from solution, down here you're slow. This is how effective you are at eliminating from your tissues. Here's a phylogeny representing what we think we know about the relationship among these organisms. And basically, these things all had reasonably strong K statistics. They were all significant, so there was this phylogenetic pattern associated with these traits. We also saw a pretty big, interesting trade-off that this is basically log A. This is your ability to eliminate it versus how much energy or how well you do detoxifying. And they detoxify in different ways, but the major way is expression of small, low molecular weight proteins with a lot of sulfur residues in them, right? And basically, organisms that don't efflux well spend more time expressing these proteins. Organisms that do efflux well don't bother. It turns out that these are the ones that go away in nature. These are the ones that kind of persist in nature. Now we're seeing some traits associated with patterns in the landscape, which is kind of cool. We got into the literature to see, like, what if we get out of just thinking about insects into other groups, and basically these are elimination rate constants for zinc and cadmium. All these black circles, the filled in ones, are insects. They're data from our lab, and these are all the other data that we can find for any other freshwater faunal group. We also wanted to know, might you, if you had data for a handful of species, but you didn't have this one, could you come up with a reasonable prediction of what this one might do based on its position in the phylogeny? And so we got very busy. If we go back to, so I'm going to go back real quick to this plot. Notice that, like, in many cases, the relatives look alike, but there's some cases where they don't. Like the hydrocytes, the hydrocycidae, they're really different from one another. The ephemeralids, these are really different from one another. And so when we sort of parlayed these early findings into an NSF grant, we said we were going to focus on these two groups because they were the ones that were the most problematic here. They're also practically really species rich, and we could get lots of species if we went to the Smokies and clean habitats and found them. So we kind of hurt ourselves at the outset by looking for phylogenetic signal and looking for basically being able to predict things on these groups that we already know at the outset were pretty variable. And it turns out that they were pretty variable. They occupy different spaces. So there's this, these guys live here, these guys live here. But we couldn't pinpoint and make really great predictions based on where they were in that phylogeny. So once you get too close, the pattern starts to sort of dissipate a little bit. So Anyway, so this is sort of, the first bunch of years of my time here at State. And while NSF is where the money comes from, sometimes EPA is not. But in terms of wanting to do work that makes a difference to the way we actually regulate and manage chemicals in the environment, like I kind of really want to stay in touch here and really want to do work that makes a difference in this regard. And so there's two very different sides of the Clean Water Act. One of them is focused on toxicology to establish basically water quality criteria. These are numbers that represent what we say as a society is polluted or acceptable or not. This is a limit. This is like and these have a lot of ramifications out in the world because they will affect discharge permits. They will affect commerce in a big way, but they are really important societally because they're saying what is acceptable. And they are made by compiling the results of single species toxicity tests. And the way of creating a number here was developed in 1985. It's not been changed since then. Right? The other side of the equation is that there's this part of the act that requires states to report on how are your streams doing. And so basically these ecological surveys assess biological integrity, but they're all observational studies that have no inferential power whatsoever. Right? And so just sort of saying in a different way, one side of the Clean Water Act is basically you have a relatively small number of test species available that are supposed to be used to make inferences across all of freshwater biodiversity. And then you're monitoring communities in nature, and basically the natural species assemblages are these big pool of available species. And generally speaking, when you're in flowing water, you have very little overlap between the species that are in your data set and the species that are in your system. So you have these pond and lake dwelling animals that have been used as lab rats, but none of these flowing water animals are represented here at all, or rarely. So in the case of metals, there's actually a fair amount of toxicity data because mines are often up in the hills, they're often in streams of low water that are dominated by insects. And so people were interested in understanding, well, how much cadmium, copper, zinc, what have you, does it take to harm these things? And so, unfortunately, these things don't do, these tests would indicate that insects are just tolerant, that they don't care about the metals. Then you go in nature and you look at the communities, and the communities tell you metals are really putting a hurt on biodiversity here. And so there have been commentaries and papers addressing this, including some from ourselves. And then we compiled over the years just a fair amount of work to have the confidence to basically say that the normal testing, the normal way we do things is pretty much irrelevant. We kind of came up with 4 reasons. And so I'm just going to really quickly walk through some of the things that we learned in our lab to sort of support this kind of bold statement. And one thing is time, right? So a traditional toxicity assay that would be used to generate a water quality criterion value is 96 hours. And we had all of these uptake and depuration kinetics. This is for cadmium. So we measured all these different species. So each point represents a different species. And basically, armed with those kinetics, we could infer how long it would take an animal to sort of reach a steady state concentration in this environment. And by and large, most species need a lot of time relative to 96 hours, which is about a year, right? So in terms of we didn't give them adequate exposure to evaluate the possible harm, okay? Reason number 2 is that the mechanisms of toxicity that are sort of the sacred cows of metal toxicity for folks who work with fish and crustaceans, they actually don't work in insects. And the idea here is that metals target or dampen the transport of physiologically important ions. So an example of this would be that calcium transport is known to be negatively affected by, say, zinc, cadmium, or lead. Sodium transport is known to be negatively affected by copper and silver. Increasing calcium concentrations protects aquatic organisms against the toxicity of these things, and so our water quality criteria are tailored to understand that aspect of it. But in our hand, so in a fish, it's basically like this. So here's your control animal. As you increase your calcium, you basically hear the kinetics of uptake, and here are your controls. And as you meter in different concentrations of cadmium, you're just suppressing this calcium influx. You can measure low calcium in the blood of these animals, and that's what's supposed to make them sick. In our hands, calcium is very modestly kind of protective, but not really. A 43-fold increase in calcium. Did a little bit here, not statistically significant there. A little bit of protection, but not much. But the real test is basically to ask if exposures to these metals, which are here on this axis, basically orders of magnitude increases in cadmium or zinc. What is this doing to calcium influx rates? These are different species. And occasionally we get a drop, but we're almost stimulating some transport. We're either not affecting it or we're actually stimulating it, but we're not suppressing it. Same thing happens with silver and copper. If you have enough silver, you can almost abolish sodium transport, but this is a lot of, so this is more than you're going to find in the environment, right? And so, but we also get stimulation. So controls here are in black. Here are different levels of silver. Basically, you get some stimulation transport. So we're supposed to see suppression. We don't get it. We get nothing or stimulation, except when silver gets really high. So the sort of sacred cows of how do metals cause toxicity acutely anyway don't sort of match, and that helps us understand why in the toxicity test the animals don't get hurt. It takes a lot of metal to make them sick. Reason number 3 is that dietary exposures are really important, and our lab spends a lot of time on this. My first master's student here, Katrin Martin, published this paper showing that they may receive the majority of their tissue concentrations from their food rather than from water. We have subsequently shifted to understand, and this is with predators, so this is if predators don't apply to eating other invertebrates, but we've shifted our attention to periphyton. Periphyton is the stuff that makes you slip when you walk in a stream, you know, that slippery bottom, right? And these are biofilms, really complex. Typically in stream environments, they're dominated by diatoms. Diatoms are really nutritious. They got great fatty acids in them. And there's like the preferred food source for lots of things. But there's fungi and lots of bacteria and greens and green algae, but mostly it's diatoms. And we've learned over the years, working with several metals, that metals concentrate in these biofilms in a huge, huge way. And so we've published a bunch of papers, you know, quantitating the accumulation of metals in these base of the food web here, and then we subsequently would feed them, you know, to insects, and with label or not with label, we look at the transfer of these materials from the food into the tissues. Basically, food is really important. So, next question is that, well, does it matter? Are food exposures potentially harmful? We showed that the water ones didn't really do much. And so what we wanted to do is we wanted it to be a fair fight. We wanted to do a side-by-side comparison, basically control the dosimetry of the animals so that they got similar concentrations of their tissues, whether exclusively from water or exclusively from food. So basically we did this 2 times, experiment 1, experiment 2. This is the concentration that we got in the tissues of this mayfly from water only. And this is the concentration that we got in their tissues from diet only. So we have a fasted control group and a fed clean food control group. We have labeled cadmium in here, and so we can quantitate what's in the water, what's in the food, what's in the tissues of these animals. So we did 2 times. Did a reasonable job of making them about even in each different experiments. And we're evaluating the sort of biological activity here through the activities of 3 different antioxidant enzymes: catalase, glutathione transferase, and superoxide dismutase. This white line is basically the control. This is experiment 1, this is experiment 2. And basically, as these guys acquire tissue concentrations of cadmium, they did nothing to the activity of these important antioxidant enzymes. And I got it from food, identical identical tissue concentrations, suppression of catalase, GST in this one case, superoxide dismutase in both cases. So this is sort of against the sort of normal thinking in metals toxicology, where people assume that the water is the most toxic thing, and that the diet is less so. In our funnel group, it looks like it's different. And so we think that these tests fail because the test durations are insufficient. The assumptions of the mechanisms aren't really supported from dissolved exposures. The dietary routes of exposures are ignored, and they may be challenging. Actually, they may be more challenging than the aqueous exposures are. I would argue that they're probably all challenging to some degree, but the food seems to get them more so than the water does. So I mentioned earlier that the methodology for establishing these legally binding numbers, these water quality criteria, were established in 1985, and EPA is actually considering maybe modernizing this process. And so last fall, they had an invited expert meeting where they said, we're going to take abstracts from anyone who wants to tell us how to what we should think about in modernizing this process. And I was really lucky that they accepted my abstract. I was able to go there and tell them what I thought about some of these things. And I'm actually working on a paper with Sandy Loma now and another prominent scientist, Will Clements, where we're sort of trying to make a case for broadening the data that's used in the development of these numbers. Because right now, if you did a test and you used dietary exposures, EPA would throw it out because it wasn't done according to the 1985 methods. They don't even consider any field work. Like no monitoring data, no observations in the field. It's only single lab toxicity tests. And we're saying, we know a lot more than we did in 1985. We should use all the information that we have to make more sound rules. And so I mentioned also that these two different sides, the ecology and toxicology sides of the Clean Water Act, don't really communicate with each other very well. And basically, each side is imperfect, right? These individual tests lack ecological relevance. Again, they ignore diet. And the data sets are just dominated by fish and crustaceans. There's only one really go-to culturable taxa for toxicity testing. This was this midge called Chironomus, which is like totally bulletproof. It's really hard to kill this thing. And it's the only thing that is really, people have in culture to do toxicity tests with. And so water quality criteria only need one insect in the data set to pass a number, despite the fact that in the communities, like 95% of the species that are there are insects. So the data is dominated by fish and crustaceans. So on the other side of things, the ecological surveys can only really tell us things like the system is good or it's bad or it's ugly, but they can't really tell us why, like what's causing it, and it's, again, dominated by insects. And so the faunal groups that are being used in these two different ways of sort of thinking about protecting water are completely different. So one idea was to sort of try to bridge these two things, so try to bridge these two sides, be able to speak the language and be familiar enough with what the ecologists were doing, what the toxicologists were doing, that I could communicate with both of them. And one of the things that we also did is we decided that we could basically develop a mayfly as the new insect lab rat to get rid of that old, really tolerant midge, replace it with something that at least represents a sensitive faunal group. Mayflies in general tend to go away first in a lot of contaminated scenarios, and so there's some aspects of this. So why no insects in the lab in general? Like when I started doing this work as a student, I sort of approached people about working with insects in the lab. It's like, you can't do it. Like you can't hold them in the lab. You can't breed them. And there's a lot of reasons for this. One of them is that almost all insects die of diapause, meaning that their eggs, for the most part, wait it out and just don't develop, and they wait for good temperatures. And so some have a winter diapause, it's just too cold to develop, so we're going to wait till it warms up. Some have summer diapause, it's too hot, I'll die if I hatch now, I'm going to wait it out until it cools down, and then I'm going to hatch. And so manipulating the temperature regimes to break diapause in eggs is a real bear. This particular species doesn't. Also, insects have all these weird cues about mating and ovipositing, laying eggs that we know nothing about. And so for sexual species, like how you provide that in the laboratory, like no one has really figured it out yet. These guys, gals, are parthenogenetic. There's no males at all. Never been a male seen. It's not like daphnids, which can do it either way. These guys are obligate parthenogens. And they're pretty highly fecund. The animal's well fed and reasonably okay temperature. She'll lay 1,200, 1,400 eggs. These are all clonal animals. You can put them in the refrigerator and they will just sort of develop real slowly. And then when you pull them out for an experiment, you get them to hatch for you, you get some good sample sizes. And we got in, early on when we first got the 454, we were able to do something of a transcriptome. We recently kind of added a little bit to that with a Illumina run, and we're just about to get started on our first RNA-seq experiments. But we've been actively developing a suite of genes that we're interested in exploiting for different projects. So, so to kind of take this lab biology, but also make it useful and practical, we like this thing. And just as a means of history, it started here at collaborators of ours at the Buchwalter Research Center. They shared it with us. And since we started popularizing it for use in toxicology, it's gone to Illinois, it's gone to a national lab in Ohio, it's gone to another national lab at USGS in Colorado, another academic lab in North Dakota. So it's starting to be used more and expand. And so we're hoping that that continues to occur. And finally, just with the last couple of minutes, we do other things besides metals, and really over the last 5 years or so, we got very busy thinking about temperature, and it took all of that time to finally secure an MSF grant on it and several failed attempts. But I led this collaborative effort with the Buchwalter Research Center, with Vern Sweeney, Chuck Hawkins at Utah State, Gagi Davidowitz at University of Arizona. And the general idea here is that we're really interested in trying to understand the physiological basis for thermal tolerance across species. So these animals, in monitoring context, are used to evaluate water quality, but we have no way to tease apart the influence of the temperature on these communities versus other water quality aspects of these communities. And so the idea here is that the folks at the Stroud Center have the capacity to rear insects through their whole lifespans. And so from egg to egg, under different thermal environments, we then get the tissues of these animals. And our lab is doing things like measuring oxygen consumption in them. We're doing some metabolomics with RTI, and we're doing some mRNA expression work in Triangular Fur. We're about to do an RNA-seq experiment in these animals that have been raised in different thermal environments. That's the metabolomic profiling data from insects raised in optimal versus suboptimally warm environments. So we're really interested in the hot side of things. We're also interested in sort of plugging these into these ecological niche models, which are describing relationships between species occurrence and temperature. And we're trying to hopefully give them the right measures on this axis to be looking at, because it's unclear whether it's mean summer temperatures, mean annual temperatures, degree days above X. There's not, we don't know what the right thermal metric is here to best marry the field data to. In Triangular Fur, anyway, well, let's leave there. And so, just to close, ultimately, stewardship of our planet is in our own best interest. We don't really have the ethic to just preserve things for their own sake. It has to come back and benefit us somehow, right? But it is good practice, right? Good clean water is like money in the bank for a nation. I mean, it's important. And honestly, the things we do in the environment, short term, they ultimately come back and haunt us or our offspring in the long term. And so lots of people at bank, early mentors who let me work with bugs even though they had no idea what I was doing. And so that was really great. I mean, if I hadn't had the opportunity to start this, I wouldn't be anywhere. And Sam Willow at USGS as my postdoc mentor, and Jerry was assigned as my faculty mentor when I arrived here, and has gleefully ripped apart many of my manuscripts and grant proposals over the years, and I appreciate that. I appreciate that because it helped the process out a lot. Had great students and a disproportionate amount of awards won by them. Some very good publication records for them. Postdocs, a bunch of collaborators, including some of you guys here in the room. Funding has come from a variety of sources. NSF mostly, a little bit of EPA, but not as much as it should be. USGS, WRI, EPRI, and Rob Center, who's actually helping us out with a pilot project. Partially removed from water. Anyway, with that, happy to take any questions. Thank you for your attention.
Questions?
Yeah.
So, you said that you're trying to change the testing stuff. So what about the new hospital? I don't know if you follow that, but the Toxic Substances Control Act, right? So they have a lot of language in there about new tests and new, innovative approaches, and it's almost like the burden is on the community to convince them, the regulators, the powers that be, that you can do something.
Yeah, so I don't know exactly what the strategy is for actually, I mean, the reason why it got changed was because we had fallen way behind in the testing of things using commerce. I mentioned there's water quality criteria. We have 58 water quality criteria on the books right now, 58 chemicals. There's something like 89,000 chemicals used in commerce. Maybe 7,000 of them are common or widely used, right? So 58 out of 7,000 is a pretty bad batting average. And partially because TSCA, when it was written, Toxic Substances Control Act, didn't really require a whole lot of testing. It's a pesticide. It's under FIFRA, requires a lot of testing, but for most things it doesn't. So I don't know. I haven't read enough about their strategy, how they're going to change it, or whether they're going to demand industry to pay for some of this testing. That seems like a reasonable thing. That's what the Europeans do. We don't make them do that here so much. Jerry, any ideas? Look at that. What the strategy is for TSCA-related testing? No. Anyway, yes, I can't answer. I know that there's been, the TOX 21 and this whole high throughput thing has, gained a lot of momentum. And there are certain places where there's some work on the AH receptor and its morphology that suggests differential sensitivity, that you could almost predict it based on the way that receptor looks. And that would be an interesting thing to sort of combine with phylogenies and see you could make some predictions that way. Yeah.
So I understand that global warming is a giant problem, pollution is a giant problem, but in terms of freshwater ecology throughout the world, is it clear which is the bigger problem?
Pollution versus temperature?
Yeah.
I think it's all going to vary, right? Some places going to be pollution, some places.
So averaged over the world. So if you're making budgets or doing good things, how do you divide the money?
How would we prioritize?
Yeah.
I guess I don't know how we prioritize. With our mayfly lab rat, its performance falls off a cliff really quickly. So it's not a gentle slide from reasonably good performance to just over time bad performance. Like a couple degrees will crush them over their developmental period. So, I mean, what would happen if you just say raise temperatures is you're going to lose some species. You're going to gain other species. Warmer ones are going to be able to exploit those environments. You're going to lose the colder ones. The systems will probably continue to function. It'll just be different. The pollution, you're going to just lose the hell out of species. And so maybe I would sort of still prioritize the pollution of the things.
related to that, so is it just the dropping oxygen concentrations with the freezing temperature, and they're kind of pushed to the edge already?
Well, that's what we're exploring is the role of oxygen. Our oxygen needs met. I thought about presenting some of that work. Basically, there's a prevailing idea that thermal tolerance of ectotherms is driven by maintenance of aerobic scope. That when animals are pushed, that aerobic scope shrinks, that kind of hammers them. None of the work that we've done supports that. So if we make animals hypoxic, we can light up these genes that are hypoxia-responsive genes. When we heat them up, we don't express those genes at all. Our metabolomic profiling work doesn't show a bunch of anaerobic metabolites. It shows all stuff related to membrane maintenance and stability of fatty acids sort of stuff. Our respirometry suggests that there's, at ecologically relevant thermal limits, there's still a whole lot of scope for aerobic scope. We haven't proved that yet. In fact, we just had a paper get hammered on that we didn't do that correctly. But so far, none of the stuff that we're doing is supporting that limitation. We think it's more bioenergetic related things, like just the maintenance costs and the distribution of caloric resources to maintenance versus growth become very skewed. When they're hot, they grow really fast and the adults are tiny. And they lay very few eggs. When they're cold, they develop really slowly, they're fat and happy, and they lay a lot of eggs. So fitness consequences of temperature are pretty big, pretty measurable. But not happening through oxygen availability or supply demand issues probably. Yeah.
You showed it's not the transport ions that are killing the insects when you increase the metals. Do you have a sense of what it is that's causing that toxicity?
Well, I mean, so metals are toxic in a lot of different ways. In the short-term things that we've done, we haven't showed a disruption of normal homeostasis, but we have showed issues of oxidative stress, with dietary exposures. And that's certainly one way that metals can be problematic. Is like reactive oxygen species. Scavenging of important redox components like methane cells seems to be important in our organisms. Metals are going to bind to, cadmium, for example, will bind sulfur. It's a lot of sulfur in the active sites of a whole host of enzymes, right? So there's a lot of different ways. It's not like one mechanism. It's like lots of mechanisms that something that can be toxic. Yeah.
Dave, given that you've shown that it's the metals in the food that are important, what do you foresee in the future in terms of the EPA right now doesn't have any criteria for metals per square meter of stream bottom, right? They probably never will. They're probably going to want one for dissolved.
Right.
And what's the correspondence between dissolved metals and
Yeah.
how strong is that between dissolved and what you see in graphite?
Yeah, so we do a lot of work quantitating the transfers of metal from solution into paraffin. And so regulations are always going to be tied around the dissolved number for the most part, although there are the latest water criterion for selenium isn't so much, like how to provide this item. But it's not that crazy to think that you could infer concentrations of biofilms from concentrations of water, and if you took a more holistic view of exposures, that you could still regulate on a dissolved concentration, because that's still going to control exposure. It's just not directly. So one of the things that recently happened, one of the reasons why EPA has recently convened this meeting about modernizing criteria, is that there's this element selenium that broke all the rules about how toxicity can be evaluated. Because when you expose a fish or an insect or whatever to selenium in solution as an oxyanion, selenate or selenite, uptake into animal tissue is like negligible. So these things happen out in nature. You had here in Bellew's Lake, collapse of fisheries, right? Because of selenium. California, you had all these malformed birds in this wildlife refuge because of selenium. And we've now learned that selenium gets taken up by aquatic plants gets converted to organoselenides. Those organoselenides get trophically transferred. Egg-laying animals take that organoselenium and sock it into eggs. And so when you don't do anything to the moms, you affect their offspring. And so the normal testing paradigm of like exposing different species through water completely failed them. And then they went through the process of like, well, how do we do it then? You know, if we can't do it that way. And so a lot of science went into figuring out all those different steps, and models were made and all of this, and now there's new values that are sort of like, it could be a fish tissue residue or it could be water concentration. Of ways to link these things and ways to, like, in this scenario, it would be a different number than in that scenario. And so because EPA had success and did it, they're like, well, maybe we could actually do it right for other things, you know. And so the window of opportunity is like cracked a little bit. Like now that they've seen that they can kind of use a different approach to setting a number, that they might be more open to it. And hopefully there's just like enough weight of evidence that this dietary thing is important enough that failing to consider it is just leads to uncertainty about whether these things are protected or not. Yeah.
I had a couple questions about the community ecology of these streams. When you see widespread loss of species, do you think that they are all being physiologically affected by copper or what have you in the same way, or could it be that there are a few sensitive keystone species that when you remove them, you're going to lose a bunch of other species?
Yeah, there can be both direct or indirect things, for sure. Yeah.
And then related to that is whether you can kind of turn the question on the head, and if these communities are, if these species maintain their niche by competition, can you ask what's so special about mayfly physiology that allows them to outcompete everyone else, so long as it's cool and clean?
Yeah, I mean, I don't know why that group is so successful in clean streams and so terrible in impacted ones, but that seems to be the case. They're the most ancient of all of the groups. It's debatable whether dragonflies are older than mayflies, but when I popped up, you know, this lineage is popping in. That first one is mayflies and odonates at about the same time. The odonates seem to be a lot more robust, you know, the dragonflies, damselflies. The mayflies seem a lot more temperamental. They've survived all these different mass extinctions, so, you know, they've persisted. But they're not doing so well in this epic right now.
I think it was with selenium, you showed data where comparable doses of either water or food resulted in different effects on proteins that were involved in action.
That was cadmium.
Cadmium.
Yeah.
Which is interesting and peculiar. Is there any information, any data that indicates that carries through to toxicity? That cadmium from food is more toxic than cadmium from water?
We haven't done that exactly. We've done, so in criticizing the way the tests are run, you know, the durations and the dietary exposures, what we basically developed was a way to expose these things from like the day after they hatch until they fly out as adults, like the whole lifespan essentially. And we have showed dietary toxicity of different trace elements. We haven't tried really hard to manipulate those exposures so that we could be sure that they were overwhelmingly one or the other over about a month of their lifespan. That would be kind of challenging to do. Paraphyton takes some time to load up, but it's, you know, so when you add the food to them, the metals sorbed onto it or get into it in a big way. And so how do you keep them fed without having a dietary exposure? And the short-term thing that we did, we could do it, they could fast for the 4 or 5 days that we needed to load them. For a long-term thing, though, you can't completely isolate the exposure rows.
Second question.
Yeah.
The species, the pharmacogenetic species that you use in the lab, you talked about how in environmental toxicity testing, there are a few species that are used typically to evaluate toxicity. We hope they're representative of other species. Do you have any, and you also mentioned that the insect that is historically used is actually quite tolerant, it's not representative. Do you have any feel for whether the species you're working with is more representative?
Yeah, it is. I mean, mayflies are, well, midges are important in ecosystems too, but mayflies are often dominate in good conditions. And they're definitely more sensitive. So the argument from sort of industry is like, it doesn't really matter if the faunal group is represented as long as what's in there is sensitive enough to cover them. And that argument has sort of failed in a few places. Like we had a criteria for ammonia that was based on understanding of ammonia toxicity in fishes and crustaceans and other things. And then recently it was discovered that, well, actually freshwater mussels really can't tolerate ammonia. And so we had a number on the books for a couple of decades that was not protective of aquatic life, and then that was fixed, right? Similarly, in like these mountaintop coal mining removal sites, people are using Ceridapnia dubia in these whole effluent testing scenarios, like how bad is this water? And that species, which is thought to be sensitive to a lot of other things, is pretty tolerant to the salts that are coming off of these mining situations. You put a mayfly in there and it just kills them. You know, and so we don't, just because an organism is sensitive relatively to one thing doesn't mean it's going to be sensitive to something else. And so we're kind of trying to make the argument that we need a little bit of a broader faunal representation in the data sets to create these numbers.
Do you have any idea whether the clone of mayfly that you're using is representative of a wild population? Is there any differences in terms of sensitivity or resistance?
There are some differences in it that are, you know, apparently our clone does not have like some of the normal gut symbionts that wild ones do. So there's somebody who learned that recently.
Microbiome.
Yeah, exactly. But other than that, I mean, most insects are not properly genetic. So in terms of that in itself is like, no, it's not the most representative thing in the world, but practically it makes it feasible to culture it. You know, so we're not going to argue that it's like the most representative thing in the world, but it is definitely more sensitive than the old one, and a better choice for if you're going to use these things to be protective, you want to use the more sensitive ones. So yeah. Anybody else?
What species are ubiquitous?
East Coast, back down to Georgia. And there's lots of different clones.
Any other questions?
All right.